This conference will now be recorded.
Okay. So today we're going to go through part 2 of the SARS-CoV-2 data submissions training. Today we're going to go over, yesterday we went over just a general review. Today we're going to go over how to determine which samples that you're going to select for submission, how to review them, and then prepare your multi-FASTA file for submission. So just a reminder, this is part 2 of a part 5 or 5-part training series. So today we'll do sample review, FastaPrep, and tomorrow we will do, we'll go through the submission, so the FASTAs that I'll show you how to prepare today, show you how to use those tomorrow to use the web portals to submit to GISAID and NCBI. So by the end of this week, you'll be able to submit the consensus assemblies and sample metadata to GISAID and to NCBI Biosample into NCBI GenBank. And then again, as a reminder after that, if your lab would like to set up individual calls with me, we can go do a walkthrough one-on-one or with a group from your lab to practice using your own data to submit, troubleshoot, make sure there's nothing going wrong. And then after that, I'll go ahead and set the days and times for part 4 and part 5 of the training. So for today, just an outline, we're going to go through how to review your samples, batch those samples for submission, just talk about strategies of what's recommended, of how to submit and how often, how to assign your public repository names to your samples, because they all have to be renamed to a particular format, suggestions or recommendations on how to organize your metadata, and then how to use Hypergator to generate your formatted multi-FASTA files. I showed you this yesterday just to kind of review what we're focusing on and what we're trying to submit here. So from whatever pipeline that we're using to analyze your data, the vast majority of them should be outputting roughly the same thing of a consensus assembly. You'll have your associated sample metadata in your FASTAs and your BAM files that are all the focus for submission. And for parts 1 through 3, we're focusing on that consensus assembly for submission. So again, I showed this to you yesterday, just kind of outlining the entire submission process and what's involved for submitting those consensus assemblies to GISAID and NCBI. Today, we're going to focus on what's in blue. So we're going to screen passing QC samples for submission, show you how to screen this with Vader, which is NCBI's viral annotation pipeline, how to select the samples from that for submission, how to organize your metadata, assign your public repository names, and then how to prepare that formatted multi-FASTA file for GISAID and GenBank using a script in Hypergator. And then tomorrow we'll go through the rest of the pipeline of how to actually submit that and what to submit. So sequence your samples, you have your data, now what? How do you select those samples for submission? Well, whatever pipeline that you are using, you should have some type of indication of which samples pass QC or not. There's different recommendations out there. Yesterday, I provided a link to some recommendations that were pushed out by EPHL and with guidance from CDC as well as some other state labs and bioinformaticians. But if you talk amongst the states, people use different thresholds in their labs. For example, here in Florida, we submit anything that has greater than or equal to 80% genome coverage. So that basically we have a consensus assembly that is covering at least 80% of the reference genome, with each site has a minimum of at least 100x mean read depth. Other labs I've heard do 50% genome coverage and do that. We might, here in Florida, lower ours and submit more because some data that is a complete genome will be used for some purposes, but other data, if it's not a complete genome, but there's still complete genes or CDS regions in that genome that are submitted, those are still useful for other purposes too, especially, let's say, if you have a full spike gene. So it's still recommended to submit those samples. Some of them, if they have less genome coverage, they might not get a lineage assigned to them if they don't pass Pangolin QC. So there's a number of different factors there, but you can still submit data, as we've seen some labs going down to 50%. But here in Florida right now, what we've been doing is 80% 100x mean read depth. And APHL is about to update their recommendations with updated guidance that hopefully should be coming out soon. And we can also discuss that later if you have further questions about what those new recommendations are. So we batch our samples based on whether they've passed QC or not, and then also based on if they passed Vader. So Vader, I'm going to go into detail in the next couple slides exactly what is Vader, what is it doing. Basically, when you submit your samples to NCBI and GISAID, they both of those databases, they have curators and they screen your data to make sure that there's no errors and that it's acceptable to them and meets their criteria so that they'll include it in their databases. NCBI's tool that they use, that every single sample SARS-CoV-2 genome that gets submitted to NCBI is run through their pipeline. It's called Vader. It's a viral annotation pipeline. It's open source, so we're able to run it ourselves so that we can pre-screen our samples so we don't waste our time trying to submit a large batch of samples and a bunch of them might fail or get flagged. GISAID has slightly different criteria for submission, and they have not released their viral annotation pipeline open source. I believe they might have some type of web portal I've heard of where you can upload your samples and they get screened that way. But they haven't released that open source, and so that's caused a lot of frustration among a lot of the labs of that some samples might get accepted by GISAID, some samples might get accepted by GenBank, and not by the other one, vice versa. There's been some recent updates that I'll discuss that have made this much easier, and there's more consensus among both of the databases so that you're almost guaranteed that if you screen something and if you submit it to GenBank, it will be accepted by GISAID. And they've added different features to indicate, like on GISAID, they're called confirmation options, which I'll be going over in the training tomorrow. I just wanted to make you aware of that just because one database accepts one genome does not necessarily mean the other one will. But screening the samples yourself with GenBank's pipeline is a good start so that you're not wasting your time trying to remake FASTA files and try and figure out why something didn't get rejected. That's been a huge bottleneck in this whole process, is this part right here in the submission process of trying to deal with samples that get flagged. So the recommendations that I'll show you in this training will reduce that a lot, and we're seeing significantly fewer rejections or flag samples. All right, so you've identified which samples have passed QC. You're going to send them for submission, but you need to review them first. Some pipelines, so for example, if you're using our FLAC SC2 pipeline on HyperGator, Vader is part of our pipeline automatically, so they've already been reviewed. So if you view the report.txt file, there's a column in there for your QC flag. It's going to say pass or fail. Pass means it has greater than 80% genome coverage and greater than 100x mean read depth. Fail means that it did not meet either one of those two criteria. The Vader flag will either say pass or review. Pass means that it passed Vader pipeline and it's ready to go for submission, and it should not be flagged, at least by NCBI, for submission. More than likely, it shouldn't be flagged by GISAID either, but we'll go over that in more detail tomorrow about some of GISAID's criteria. If you're not using our pipeline, some other pipelines do include Vader, some not. I think there's an option on the secret pipeline that allows you to select whether you want to run Vader or not. I don't believe that the Dragon pipeline, the Illumina Dragon coded lineage app, I believe it does not run Vader. So if you're using that pipeline, then you'll need to pull in your assemblies from BaseSpace after running them through the Dragon pipeline, and then run Vader separately, which I'm going to show you today. I have a script for you that you can run them, and it will give you a report whether with the sample ID and says whether it passes or reviews, and that's how you'll select your samples from there to move forward with submission. So before we get to that, before I'm going to do a live demo of how to run that script on HyperGator, I'm just going to go give you a little bit more information and provide some links regarding NCBI's Vader program so that you can go look up more information yourself. Some of the more detailed information about different types of alerts you might get will be more relevant for the Part 5 training that I'm going to put together of basically when you get a flag sample, what do you do with it and how can you review it? How can you confirm whether a mutation is real or not, or whether it's a sequencing error? We'll go over all of that in Part 5. There's just a little bit more information involved with that. So NCBI's Vader stands for Bioannotation Definer. This is just a quote. I thought it was a great text just from their GitHub site that explaining what Vader is that it's a suite of tools for classifying and analyzing sequences, basically using reference models for a number of different viral genomes. So they use it not only on SARS-CoV-2, but other viral genomes as well. So when you submit, if you sequence norovirus or, for example, like dengue, it'll be screened with Vader 2. They just use different models for that to evaluate the annotations for that. So the code for Vader and documentation can be found here at this link, and we'll go to it in a second, and I can show you all the different information there. It's really well documented. And then I've also provided a link here for the list of Vader Alerts. And basically, these are going to be any kind of errors or flags that get detected in your sequence. That'll be more relevant, again, for part 5 when you're going to dive into what those errors mean and how to confirm those. But let me go to just their documentation right now so you can see. So this is their GitHub site where you can find the software. You don't have to worry about installing or anything. That's already installed on HyperGator. Scripts are already written. I'm going to show you how to run it. But in case you were curious and just wanted to know more information, they have a specific link here for SARS-CoV-2 annotation using Vader. So if you click on this link, they have all this information and documentation about how to annotate your sequences, the different models, different updates to alerts, etc. But again, I have the most relevant information on the slides. Make the slides really quick. And related to our uses here. And again, you don't have to worry about trying to define models or flags in the when you're running it. That's already been determined for you in the script on HyperGator. I'm just going to provide this link here, but we'll come back to that on part 5. But I'll just show you that when you do go to that link, it provides, these are all the different alerts and errors that you can get from Vader and why, basically all the different reasons why your sample might be rejected when you're trying to submit it. And it can be things such as, the most common ones are that the CDS has a stop codon or the CDS has a frame shift. You'll see a lot of peptide translation problems, which you'll get when you have a frame shift or a stop codon, and you might get some low feature or low similarity ends or start. Many of these are caused by ends. But again, we'll go over that in more detail on part 5. Okay. So we're going to go through a demo right now on HyperGator, and I'm going to show you how to run this script here. It's called SC2 review Vader.py. It's a Python script, and there's an associated SBAT script with it. So again, as I mentioned in the part 1 training, that requirements to fully follow along and be able to do this: 1, make sure you have a HyperGator account. Make sure you either are comfortable working command line or that you've gone through my Linux 101 training, that you've set up your HyperGator compute environment and everything installed properly, and then you should be good. After you've gone through the initial trainings that all new HyperGator users go through, you'll be ready to go to analyze or to run these scripts. So as I mentioned before, just to repeat, if your analysis pipeline already runs Vader, you don't need to do this step. You're already good to go. You can already determine which samples pass and which ones need to be reviewed. Flag SC2 users, it's in the report.txt. So Vader is installed on HyperGator, and I'm always updating to the latest version. They make updates every once in a while. You'll need to transfer your consensus assemblies over to HyperGator if they're not already there. So if you didn't generate them there, you can either use WinSCP to transfer those files from your local computer if you have that data on a local hard drive or on your computer, or if that data, for example, is in BaseSpace because you ran the dragging for the lineage app, you can use the BaseSpace command line interface to pull those in. So I gave you an example command here that if you've gone through some of my trainings and I've showed you how to pull in, for example, like your FAST2 files from a particular BaseSpace project to be able to run an analysis, you can use that same command, vs download project, and put the project name, extension, and just indicate the file extension that you want to pull that data in, and indicate the output folder that you want to save it in. So for example, if you have FASTA files in there, you can pull in the FASTA files from that particular project, and then that's how you can just pull in that data. All right, so we're gonna run, I'll show you how to run this script. Both scripts, I'll copy them over after the training, I haven't done it yet, will be located in your blue BPHL, whatever your state or jurisdiction is, public share, and then your scripts folder. That's where I put all of your scripts, and they will be there. Okay. Let me go to okay. So I have some training data here in this folder, and I have 5 different assemblies that I want to review. So, for example, these are assemblies that, and I just assigned them names. These are assemblies that I've already run through my pipeline, and I've seen that they've passed our QC threshold. I would like to submit them, but I need to run Vader just to make sure that they don't trigger any flags or alerts, and then I can see them. So what I recommend, I've already made these 2 folders up here just in case, because I'm going to run them, and that way we don't have to sit here for like 5 minutes. Like the Vader should only take like 5 minutes or something to run on these, but that way we don't have to sit here for 5 minutes, and I can go to this to show you what it looks like after it's run. But basically what you want to do is go to a space where you're going to do this on HyperGator in your user folder, make a directory. I always recommend making directories if you're doing an analysis of any kind to always make a directory with like today's date, and then any other information that describes what you're doing in there, so that when you can go back and know exactly what data is in there and what type of analysis you did. So we're just going to do simple. I'm going to go in there. Thing in there because we just made that directory. So the first thing you'll need to do is copy over. Well, one, make sure you've copied over, transferred over your data. We have that here in our training data folder, but this is where you would use, for example, if you're pulling in your data from BaseSpace, use the example that I gave on the slide to pull in the data that you need so that you have it. So assuming that you have your input folder with all of your assemblies ready to go, then, and this script right here requires that the assemblies be separate. If you have trouble, we don't use the Dragon for the lineage app here. I have some old data from it from a while ago to play around with. I've talked to some of you in some states that use it to make sure to test out some of these scripts to make sure that it's compatible and works, and it does. The next script in the next part of the process will make a FASTA file that will take in the concatenated FASTA file that I've talked to some of you about, as well as separate FASTA files. If you require or would like this script to work with a concatenated FASTA file, just let me know and we can provide that feature. But I know that there's been some updates to the Dragon pipeline in the past, and I just want to make sure that this is compatible with everyone. So we can go back and forth and make any edits just in case there's something that I am unaware of, since we don't use that pipeline. Okay, so we're going to copy over the script. So these will be in your whatever your state is. You would do that, and then the name of and then you would start typing in the name of the script. I have them somewhere else. I just want to show you that. They will be in your public share scripts folder. And just copy them into your current folder. Okay, so you have your scripts, you have your data in another folder. You can choose to put your data in your input folder in this one. It'll work either way. I'll show you how to indicate how to specify your input. You don't need to touch the Python script, but you will need to open up your SDOT script. So remember, your SDOT script is what is used to submit your job to the scheduler of the HPC, and it also provides the command to run, so it's going to run the Python script that's in this folder. So use a text editor. I always train y'all how to use Emacs. That's what I'm most familiar with, but use a text editor to open up the SDOT script, because you'll need to make a couple edits. So I have your command, your Python command here that will be running. So it's Python, the name of the script that's in your current folder. we'll edit this because I have it, the first argument here will be your input directory where your assemblies are, and we'll edit this to reflect where mine is right now. You do not need to edit this. This is just indicating which version of Vader that we're using for this. And then the other one you'll need to edit is your FASTA extension. So basically, what will your sample be splitting on so that we know how to calculate or accurately capture your sample names. And by that, I mean, so when I listed out my assemblies, you can see that my samples are named with this structure. So I have my sample name dot consensus dot FA. So I need to make sure that my FASTA extension is dot consensus dot FA so that it can accurately capture that sample name. If yours is underscore consensus dot FASTA, you need to put that here. I think that might be one of the extensions in the Dragon pipeline. Or if it's just dot FASTA, just put dot FASTA. Whatever it is, you'll need to edit that to make sure that it's properly recognizing your sample type or your file type. So the first thing that you'll need to edit is the mail user right here, and enter your email so that when the job finishes, you get an email notification that the job finished, whether it's completed or failed. And we go over this in our Hypervader trainings. I'll put in my email. And you don't need to modify any of these features here. So as I mentioned, it's using Python to run this script. Our first argument is our input directory. Mine is in the previous folder back, so I'm indicating back to the parent directory, and it's called training data. Okay, that's where my input classes are. I don't need to touch Vader, it's running version 1.3, and my FASTA extension is .consensus.fa. So save it, close. Okay, so we should be good to go now. Submit, so we can do SBatch, submit. And you can see here that it has submitted my job. So when this finishes, and Vader for this number of samples just takes a few minutes, and you should get an email notification either whether it passed or whether it failed. If it failed, you're going to get some log. Here we go. It hasn't run yet. Oh, you can also use, make sure to remember to use the SQ-A BPHL umbrella, and you can see the current jobs that are running for anyone that's using our BPHL umbrella resources. So that's the 150 NCUs that you have access to. And you can see it hadn't queued the run yet, but let's run it again. And it still hasn't queued. Sometimes it takes a minute to queue, but when it starts running, you'll see that runtime. But it just takes a few minutes to run. And then I'm gonna go so that we don't have to sit here and wait for it. I already ran this last night. So if I go in here, this is what it looks like when it finishes. So I have the scripts that I'd copied over. I had, in this case, in my example, I had put my assemblies in a directory just currently in here and specified that in my SBatch folder. So my SBatch file, so my SBatch file looked like this. I had assemblies in, basically just indicating where my assemblies were located. So when the run finishes, the main file that you're looking for is this one right here, Vader report. So if you open it up, you can open this up in Excel. If you can transfer this to your local computer, it's just a tab separated file. You'll see that you have your sample ID and the Vader flag, whether it passed or reviewed. So basically you can use this information to now batch your samples for which ones you're going to submit, because it's recommended that you pass or that you batch your samples and submit them on at least a weekly basis to get them up into the databases as soon as possible, because it takes longer. Anything that's a flag sample requires manual review, and it takes up a significant amount of time. And for most labs, there's no way to keep up with those. There's always going to be a backlog. So in order to get the most data in the databases as fast as you can, so that it's as useful to the public health community, we want to make sure that we batch our samples by only submitting or prioritizing, I guess, the samples that pass our QC and then also pass Vader. Focus on those first, get those submitted as fast as you can, and then as you have time, you get more help. So I know some labs have the PHL genome specialists now to help with that, to go back through with their reviewed samples. So given the results from this script, we can see that, okay, we're going to prioritize these 4, and we're going to go ahead and prepare those for submission and hold off on this one. And when I have time, I'll go back and review to see what that alert is. These two log files here are just the error log and the output log from your job. So if for some reason this failed or there was an issue, you can open up these files and there'll be some additional output that can help you troubleshoot. And of course, I can, if you're having trouble trying to troubleshoot, I can always help you with that. Trimmed assemblies, this is just a directory just from the, you don't really need to look in there unless you want to. It has your assemblies, but they've been trimmed using the Vader trim script because it requires some trimming of ends off the ends of the genomes in order for Vader to work accurately. And then this is a folder here. Once you are ready to review those samples to see what those errors are, you can go into that folder there, and it contains the one of the output. There's a ton of outputs from Vader, but this is the most useful one in order to help with the sample review. This is something we'll go over in part 5 of the training, but I'll just show you really quick what it looks like. Basically, here are all the errors or alerts that got triggered from the Vader run for this sample. So we can see that there's an indication of a possible frame shift, and then usually frame shifts then trigger a bunch of other alerts based on how it may have affected the translation of that CDS. But we'll go over that. Those are more for your information right now, but we'll use those for the manual sample review in part 5. So just to repeat, the main output is this Vader report.txt. You can choose to save these. You can transfer them over to your local computer using WinSCP. It's just a tab separated file, and you'll have everything. So from there, you can then decide which samples you then want to submit. Okay, we'll go back to my slides. So those scripts will be located in your public share scripts folder. Okay, so now we know which samples, in this case for this example data, we're going to submit or prepare for submission 4 out of 5, and we'll hold back that review sample. So next we need to assign our public sample names for submission. And log everything that we want to submit. So I'm going to provide you with an Excel template. It's what we use here in Florida. You don't have to use it if you don't want to. You can use whatever you want. There's also a Phage community Excel template that I can share with you if you're interested. But basically just make sure you have some kind of way to log all of your submissions, keep track of them, keep track of all of your sessions, keep track of all of your flag samples, etc. So I'll show you what we use here in Florida. You're welcome to use it, and I'll be providing that, and it will be up on GitHub too. And I know I've already shared it with some of y'all too. So first, I do want to go over why all the samples have to be renamed. So as you saw from my examples, they all just had, I just gave them example names, whatever. Usually ours stay with our lab session. Whatever our lab session was, they go through the whole sequencing process, and then after that, we have to rename them upon submission. So for GISAID and NCBI, all of your samples will have a base name, so a base public name, which is this here, and then there'll be a different prefix depending on the database. So your sample name for GISAID will start with this, and then it will end with this. And then your sample name for NCBI will start with this and end with this. So this is to make sure that there's consistent naming, and you also get a little bit of metadata from the sample name across, and you can also then link your sample names back from GISAID to NCBI as well. So GISAID is just H519, SARS-CoV, or NCBI is SARS-CoV-2, and then this is whatever your host is. The vast majority of the samples you're ever going to be sequencing are from humans, so you put human. If for some reason you get a request to sequence samples from a type of animal, and it's a different host, just be aware of that and make sure if you do submit that data to change this from human to whatever your host was. Then you have your public base sample name. So to further dissect that, it's going to be based on the specimen, the actual specimen that's part of the clinical specimen that was tested and where it was collected. So the country is going to be based on country of sample collection, not where that person's from. If you have an international case or anything, it's based on where that sample was collected from. Same thing with the state. Whatever state you put there, it's wherever that sample was collected from. All of ours that we sequenced were collected here in Florida, so every single one of our samples starts with USA, Florida. after the dash, and it has to be in this format too, with the forward slashes and the dashes just like this. So in the middle between the two dashes is going to be your lab acronym. Just keep it the same. Whatever you choose to submit your samples under, we're BPHL, that's our standard lab acronym, so we use BPHL. You'll notice if you go to GISAID or NCBI, CDCs say CDC. The next number there, this is either your sample ID, sample number, whatever you want. CDC with their sample IDs, you'll notice they do it based on all their different contract labs. So like all the Quest diagnostic samples, like right here, we'll start with like QDX. Lab core, I think, is LC, and then a number, and they do it based on that. Here in Florida, I'll show you how we rename ours. We just basically start with 1 and then keep on going. That way, it also gives us an indication too of how many samples that we had submitted. Then the last part of the sample name is the year. This is not when you sequenced it, and it's not when you submitted it. It's based on the sample collection year. So because this becomes the virus name or the virus strain, so you want to indicate whether this is a 2021 virus or a 2020 virus or soon to be 2022 virus. So if you were to go back in your freezer and start sequencing some 2020 samples and submit those, make sure that they have the 2020 name. If, for example, and I did this when the new year rolled around and I made a mistake, and GISAID could see from our sample collection dates that they were 2020, and I accidentally didn't change it and left it as 2021. When the new year rolled over, they automatically fix that for us. But just to be aware when you're preparing your submissions. Okay, so before I get to the FASTA, I'm going to walk you through the Excel template that I'm going to share with you and what we use to organize all of our submissions. And again, you can, if you have something else that makes more sense to you, is more clear, use that. But I'm just showing this to you in case you need some ideas or need an example of how to organize everything. So we keep track of all of our submissions in Excel sheets right now. Hopefully that changes to more formal databases in the future, but right now it's in Excel sheets. I keep tabs, you can see down here, for everything that I've ever submitted. I batch everything based on 2020. And again, that's not based on sequencing year when I did the submissions, it's based on collection date of the sample. So these are all of our 2020 collected samples, our 2021 collected samples, and then I'm making a tab in 2022. So to keep everything submitted. I will also state this before I forget, that we also batch our submissions based on data type. So we've started doing some CLIA labs sequencing, generating CLIA lab sequencing data, which uses MinION data. And based on how you do your submissions, this mainly affects the NCBI submissions. You have to do one data type at a time. So we batch all of our Illumina data together, do those submissions, and then we batch all of our CLIA labs data to do those submissions as well. And I also tend to batch based on, if I'm doing a bunch of 2020 samples, do those together at once and do 2020, just to keep it all, since they're on different tabs, just try to keep everything as organized as possible, because it's really easy to lose track of something just because there's a lot of moving parts here. So how we organize everything, the samples that I've decided to submit. So as you can see, based on what I determined were just the passing samples. So this was samples 1, 2, 3, and 5. Number 4 is review. So I'm going to hold off on that. I'm not going to submit it, so I'm not adding it to my sheet yet. This is just my sheet for everything that I'm preparing for submission, and then everything that I submit. So when I submit that later, that'll go on the sheet later. So I have samples 1, 2, 3, and 5. And what I'm going to do is I'm going to assign them. Then this is how we keep track and assign our public names. We assign them their base name here. And then I have a formula here in these cells where I can just pull this down, and then it fills that in for me. So that now I have my GISAID name and my NCBI name. I then go through, we keep separate sheets that keep track of all of our sequencing reports and all of our sequencing metrics. So I go to those sheets and pull in the relevant information. So we have, based on our collection date, our host, which majority of the time is human. You'll need the sequencing platform. I keep track of, we organize all of our data based on our base projects. So we put that there so that way, if there's ever a problem or an issue or something, I know exactly where to go back on HyperGator and find that data. So I'm not having to hunt through a bunch of different files to figure out where it is. All of these columns in this sheet are based on the columns that you'll eventually have to fill in the metadata templates when you do the different submissions, and we'll go over all that tomorrow. So that's why I have these different fields here. It's because we use this sheet to just go copy, paste, copy, paste, copy, paste, and we're filling out, because you have to fill out 3 different metadata templates for GISAID, Biosample, and GenBank. That way we have this one sheet that we can go to that has everything and just copy from it, and it makes the whole process a little bit faster. So we have which sequencing platform this was sequenced on, our assembly method. So this will be whichever analysis pipeline you use. If there's like a formal name for your analysis pipeline, just put that there. These are the assembly methods. So this is the read mapper and the program that generates our consensus assembly in our pipeline. So that's what I do there, so that people know that we use these 2 programs to generate our consensus assembly. This is the mean depth across your genome for that particular sample. You also will go over this more tomorrow. You need the reason for sequencing. CDC is asking everybody to tag their samples in GISAID and in Biosample in order to indicate whether it is a baseline surveillance sample, so whether that's a sample that you just randomly selected for sequencing to get an idea of what's generally circulating in your state or community, versus targeted surveillance, which will have a bias to that selection, whether you targeted it for surveillance for breakthrough infection, a particular outbreak or cluster of interest, disease severity. If you were sequencing some samples from hospitals for people that have been hospitalized, that would all be targeted because you were sequencing that for a particular reason other than just getting a representative sampling from your community. So they need to know that reason for sequencing, and that will go into some of the metadata templates that we'll go over to tomorrow. Then I have just different fields here to keep track of all of our submissions. So I use this and start filling this in as I'm preparing our submissions, so I can copy from here to fill in our metadata templates. But then as those submissions get completed, I then fill in the information associated with, for example, like once we get our GISAID sessions, I copy those in. Once we get our biosample sessions, I copy those in. Once we get our GenBank sessions, so that anytime someone comes back at me and says, hey, can you grab these 12 samples or provided sessions for this study or for this purpose, or the CDC is interested in these sequences, all I have to do is just go to this sheet, look up our sample number that they're interested in, and pull all those sessions for them. And so it just keeps everything nice and organized. And I also link everything to our bio project. I have fields here later on for FASTQ submissions, BAM submissions to the SRA. We'll go over those in part 4. Here in Florida, we're still working on our backlog to get those submitted because we had been prioritizing our consensus assemblies, and we're going to start submitting our FASTQs soon, but that is recommended to submit those. So we fill all those in. I also make sure to put this when I did the submission for GISAID and for, I have that over here for NCBI, so that way sometimes, usually your sessions come back really quickly, but sometimes if there's a couple-day lag or something, or again, there's an issue I'm trying to troubleshoot, I know exactly what day to go back in my notes or in other files, and I know how to find the relevant information I'm looking for regarding that submission. So let me go over to I have a extra tab here I kind of use as a scratch sheet to help fill in and format our mean depth and our public means here, because if you drag this down, it's gonna rename, it's gonna order just the year. So I just kind of keep a tab here of names that I auto-populated, so I have, that way I can drag down this row and that it will, all the numbers will go in sequential order. But I basically have a list here of numbers all ready to go that I can just copy and paste from for whatever I'm next in the list for 2020, and then this is for 2021. So if I'm filling in my next samples here, I see that my next number is 5. I go here for 2021 and click 5. I can just copy, paste, and then that way I can then just drag down the formula here and do that. So that's the quickest way that I did without having to write a macro in Excel and do that. So that's how we keep track of and assign our public names. And then I also have a little scratch area over here for the coverage formula. So it depends on what your pipeline outputs. Our pipeline outputs everything with decimal points like this. So basically I can copy over that data from our sequencing metrics reports, paste it in here. It gives us a rounded number with the X, I can copy that and then paste it in here. And that way it's ready to go in the right format for the different metadata templates. So this will be shared with you on GitHub. And then I'll now show you how to prepare your multi-FASTA file for submission, so that tomorrow all we have to do is walk through the different submission portals with our metadata templates in our FASTA file. So we've identified, we had originally started with those 5 samples that passed QC from our pipeline that I wanted to submit. I then ran them through Vader and saw that one of them got reviewed or had a review flag, so I need to set that one aside and go manually review that later. So I'm now ready to go. I filled in my metadata for these 4 samples that I'm preparing for a batch submission. So the first thing that you'll need to do is the script on HyperGator that we use to generate the FASTA file. It needs to know what it's going to do. It's automatically going to rename all of your FASTA files for you, all the headers and everything. It's going to change it from whatever your lab name is to the public name, and does that automatically for you. So you can do, GenBank has a limit, you can do 1,000 genomes at a time. So this will, if you had 1,000 that you were preparing, it would rename all 1,000 genomes all at the same time using the same script. So what we need to do is we need to prepare just an input file, it's just a tab separated file for that script and transfer it over to HyperGator so it knows which samples to rename it. So just go open up a new Excel workbook here and just copy over these 2 columns. Do not include the header, just include just the names right here. So basically your lab name and then the base name. You don't need to worry about these because the script will automatically add your prefix and create 2 separate FASTA files, one for GISAID, one for GenBank, all ready to go for you, and it reformats everything. So just go ahead and paste that in here, and then you'll just save as. Let me move it in the folder. And just make sure to save it as a navigating to the folder file. Okay. Just make sure to save that as tab limited. And I've already saved this file here, but I usually always save mine. I mean, you can save them whenever you want. It doesn't matter as long as you indicate that as your input when you're writing the script. I usually always save it as the date, so the year, month, day. I usually just give it a description like seq sub. These are the sub names for the sequences. And then I like to just say what number batch I'm doing 4 samples, a 4 batch. That gives me an idea because once you start doing a bunch of submissions, and sometimes you might start more than one in the same day, it just gives me an indication of which files belong to which, especially when you start getting your accessions back from the databases and you date them and they might be with the date from tomorrow. It's really easy to start mixing up your files if they have different dates on them. So I like to use the date, but I also use the batch so I can, because usually I'm never submitting the same number of batch samples. So. Anyway, so you save that, make sure to save it as just a tab delimited file. And I'm going to close this since I already have that saved. And then that's that. And then you'll use WinSCP to just drag and drop that file over. I've already transferred it just in case I was going to run into network issues. Like I did yesterday. And then go into HyperGator, and I'm in the same. I'm in the same spot as I was before, just in this training folder for training data. So I still have my assemblies that that include everything that I originally wanted to submit, so all 5. You don't need to worry about, you don't need to make a new directory and just copy over the 4 that you want to submit or remove that one or anything like that, because the script will know which samples you're preparing for submission based on that input file that you just prepared where you only had those 4 listed. It's only going to look for those 4 samples and format those. So you don't need to worry about any extra assemblies that are still in there, especially if, let's say you had 800 assemblies in there. You don't need to worry about trying to figure out which ones you want to copy over and everything. That's why you indicate it with the input files. You don't have to do that. Okay, so I'll make a new directory. It's called sequence submissions. So just make a place in your user directory of where you want to do your sequence submissions from, and basically this is just to make your FASTA file. there's going to be two scripts that are in, again, they're going to be in your public share scripts folder. Mine are in a different spot. The main one, so the script that's actually doing all the formatting and everything is called SC2 fasta for sub. Some of y'all on the call, I've already shared this script with y'all, but it was an older version. I made some updates to it, so you can start using the new version. And I'll go over that. Let me pull up my slides really quick again so I can just go over those updates really quick. So for this script, so basically just to repeat kind of what we're doing here and why, is that GISAID and NCBI both require just one multi-FASTA file per batch submission. So when you do your submission, you're going to have one metadata template that you attach and submit, and then you're going to have one FASTA file. So when you have like 800 samples you're trying to submit, you can't try and upload all 800 using these submission methods. And as I mentioned, all samples have to be submitted. All samples that are being submitted must be renamed to that public name. So again, that's what this file is doing. It's identifying, it's pulling from your input file, indicating which samples you want to prepare for submission based on the ones you put in that input file. So if you had all of your assemblies in that input file, including the flagged ones and the passing ones, it's just going to pull out the passing ones based on what you put in that input file. It's going to rename everything. It's also going to reformat everything. So some pipelines spit out a FASTA file that just has the text all in one line. Some of them have a multi-line FASTA file. So you have, like every 75 or every 80 characters, it makes a new line. So this script supports single line FASTA files. It supports multi-line FASTA files. And so that's some of the updates that I recently made based on some feedback from some of y'all that are using different pipelines. It also accepts concatenated FASTA files already from the Dragon app. So if you already know you just want to submit everything and maybe do sample review at the same time, you can go ahead and just provide that one concatenated FASTA file. It's a separate, and then it also will trim, if you have any ends at the beginning of your genome, it'll trim those off because GenBank and GISAID don't like that. So it'll do all of those things and then output just everything we need in that one file. So it's this one script here, it's this Python script here, along with this SBATch script where you'll specify your inputs and change your email and everything and how it submits that job. Okay, so going back. So let's open up the SBATch script. And again, first thing is change under mail user, add your email so it sends an email notification to you when the job has run. And this one, if you're only doing a handful of samples, if you've gone through some of my trainings, you don't actually need this SBATch job. You can initiate a interactive job, and you can just run this script in real time because it doesn't take that long. If you know how to do that, or I can show you in one of our trainings. Otherwise, you can use the SBATch script and submit it as a job. And but if you have a handful of samples, it literally takes seconds to run. If you have several hundred samples or 1,000 samples, it'll probably take several minutes to run, but still it's really fast. It's just several minutes. So we added our email, and then I always like to review my command to make sure I've indicated all the flags properly. So Python, telling this script, it's asking for your input assembly directory. So we need to change this because I have my data right now in this directory here. It's going to ask for the name of your metadata file. So that's just your name. That's just the file with the names. So you can do Control-Z to put that in the background. And we can get the name. Actually, I need to copy that over. I forgot. So that's the file that you would have transferred over from your local computer that we just made. Mine is in a different file. Let me copy it over. There we go. Okay. Now I have so this is just our input file that has these 4 names and these 4 names. So I need to indicate that. So if you ever forget what you named it as, you can just do Control-Z to put Emacs in the background. just highlight and let go to copy, and then do FG for foreground. There you go. And do that, and then right click and it pastes it on there. So now we have the name of our file. And then I also put this here to put your state. You can put your state name underscore like whatever your lab acronym is. That's just for when it provides the file name at the end. I'll show you just to make sure you have your state name. You can always rename the file at the end too if you forget, but we just put Florida underscore PHL. So we've done that, indicator input, and then save. And then we can then execute this, just like that. So that's going to run, and then go back to the one I ran last night, so I can just go over what it looks like when it's finished. So I had my inputs in a separate folder here in assemblies, but you're going to see your scripts from your input folder, if you have copied it in there, our name file. And then you'll see an error and output log from the job. So again, there'll be any output in there if there's any kind of error or anything. So the main output from this script is going to be a folder that's going to have today's date and then bulk submission. So if we go into that folder, here are your two files for submission ready to go. So there is your GenBank file and your GISAID. And that's the reason why I have you indicate which state you are in your name, just so that it can properly put it in your file name, which you can always rename yourself if you want to once you transfer it over to your local computer. But that way it just indicates where it's from. So I'll show you what this looks like. So for GenBank, you can see that everything's been formatted properly, wraps every 75 characters, and then all of your samples have now been renamed to the proper name using that input file. You can also check too to see all the samples that are in there. If you use the grep command, which looks for patterns, and pull out every line, basically every header line that starts with that, and then indicate the name of the file, you can see that you can see our 4 samples here. So we have 1, 2, 3, 4. And then you can do the same for the GISAID. You don't have to do this each time, just to show you. Just to see how everything's been renamed. So everything has their name for GISAID, or GISAID and for GenBank. And then all you have to do, these are ready to go, and you just use WinSCP to then transfer that file back onto your local computer, and you're ready to go for submission. Let me pull up the slides. All right, so that's it for today. I was just going over how you select and batch your samples for submission, how you run Vader is not part of the pipeline you normally use right now, how you can run Vader on Hypergator and get that report to indicate which ones pass a review, and then how you can automatically make your multi-FASTA file and ready to go for submission. So tomorrow we'll actually just walk through each one of the submission portals, and I'll show you just the different options that you can select and what they mean on Friday. And then if you would like to schedule a call with me to do this hands-on yourself with your own data, just to make sure that everything works properly with your files. I tried to include just the feedback I've already received from which pipelines are being used in our region by the different jurisdictions, different labs. I tried to write the scripts to make sure that they work with all those different formats, because people are using different things. But if you're using something that I was unaware of and the script's not working properly, let me know, and I can make those edits to make sure that it works with your script. Because some pipelines will format their FASTA headers differently, or their file names differently, or things like that. And so we'll just make sure that everything works. And then I'll reach back out to y'all later to schedule parts 4 and parts 5. And as I mentioned, all the recordings from each training, slides, and any material. So for example, like the Excel sheet I showed y'all will be available on GitHub, and I'll be copying those scripts over to your public share script folders. All right, are there any questions? And feel free to either unmute yourself or put any kind of questions in the chat if you have any questions. Okay. All right, well, we'll go ahead and end today's call, and I'll make sure and share all those materials, and I will talk to y'all tomorrow.